So welcome everyone back for the last session of our summer conference this year, and we're going to hopefully we're definitely going to end with a bang. Please keep watch your calendars for the winter conference that's going to be in February. We're really planning for this next winter conference to be really good. I say we're going to make this winter conference the best one ever. So please mark your calendars for the winter conference. We'll be sending out postings on those dates very soon. All right, so our next speaker, let me get my paperwork here. Our next speaker is Dr. Charlotte Sumner. Dr. Sumner is a professor of neurology and neuroscience at Johns Hopkins University School of Medicine. Her scientific training include neurogenetics fellowship at the National Institute of Neurological Disorders and Stroke. She cares for patients with inherited neuromuscular diseases and co-directs the Johns Hopkins SMA and CMT clinics. Her research focuses on the genetics and cellular pathogenesis of inherited motor neuropathies, with particular attention to identification of disease genes and characterization of the molecular and cellular mechanisms. So please join me in welcoming to the microphone Dr. Sumner as she talks about the gene targeting therapies for neurological diseases.
Good morning, everybody, and thank you, Dr. Patton.
It goes back if you need to go back.
Yeah, that's great. Thank you. So thank you to the Texas Neurological Society for inviting me. I've really enjoyed this meeting and learned a lot over the last couple of days. This talk's going to be a little bit different. It's a little bit more forward-looking. Where are we headed in terms of these neurodegenerative diseases and treatments for them? And I'm going to use spinal muscular atrophy as kind of an example. These are my disclosures, and I want to acknowledge some folks who help me think about other neurogenetic disorders for which we're developing these kinds of treatments. So just, high-level view about neurogenetic diseases. Monogenetic diseases in aggregate are probably not as uncommon as we typically think. Maybe about 1 in 1,100 people are affected with one of these disorders. And of course, they have distinct inheritance patterns, dominant, recessive, X-linked, and that can result in loss or gain of function of involved proteins. Unfortunately, these disorders generally cause a very heavy burden of disability and often early mortality. And although we've come enormous strides in terms of identifying disease genes, particular treatments that target these neurogenetic disorders have kind of lagged. That's changing, and it's a really exciting time in the therapeutics development for neurogenetic disorders. But I just remind everybody that maybe part of the reason treatments for these disorders has lagged is that the CNS and PNS are sort of difficult to access and have very complex anatomies. So just to remind you of some of these disorders, we all know them very well, the CNS disorders like Huntington's disease, spinal spinocerebellar ataxias, and so on. And then neuromuscular disorders, including a couple I want to mention today, spinal muscular atrophy, which of course is a recessive disease, and familial forms of ALS, which is a dominant disease. So those that I've starred here are diseases I'm going to touch on during the talk. So why have we made significant strides forward recently in terms of developing treatments for these disorders? Well, it's really because we've developed novel therapeutic platforms that target at the DNA and the RNA level. So most of the drugs we use in clinical practice target at the protein level. But now what's happened is we've developed technologies that allow us to target at the DNA level or at the RNA level. And so, there are numerous types of platforms. Viral vectors can be used to replace genes, to knock down genes, to edit genes. Antisense oligonucleotides and silencing RNAs can be used to target RNAs in various ways. And even because of the increasing understanding of the complex tertiary structure of RNA, we've even developed ways of targeting RNA using traditional small molecules. So I mentioned that I wanted to use SMA as an example of where I hope many different neurogenetic diseases will go. SMA is a recessive motor neuron disease, and as hopefully many of you know, this is a disorder that really causes proximal greater than distal weakness. It's not as uncommon as people think. About 1 in 10,000 new births are a child with SMA, and about 1 in 40 of us carry the disease gene. The genetic cause of SMA was discovered in 1995. It was actually one of the later big neuromuscular disease genes to be found, and I'll explain why that is in a minute. But it really provided a very tractable opportunity for developing genetic therapies. And part of that reason for that is the disease is due to recessive mutations of this gene called SMN1, but all patients retain kind of a backup copy of the gene, what we call SMN2, that's alternatively spliced, thus making less SMN protein, but importantly present in every patient with SMA. Traditionally, SMA has been the leading inherited cause of infant and early childhood mortality, so significant burden of disease. Just again, to remind you all, SMA has this really broad phenotypic variability. I've still, as a clinician, struck by this, that you can have type 0 disease where an infant is born weak, all the way to, adult onset disease. And I actually took care of a professional wrestler once who actually genetically had SMA, but I certainly couldn't tell that he was weak. Unfortunately, the severe forms of the disease are the most common. So SMA type 1, which is defined by onset within weeks or months of birth, no ability because of the profound weakness to ever achieve the ability to sit independently, and before treatment, usually lethal by 2 years of age. Unfortunately, 60% of patients have this severe form of the disease. So I assume all of you have heard of what has happened in SMA. In the last 6 years, there have been 3 different treatments approved for this disease, which is sort of remarkable and a breakthrough for neurodegenerative diseases in general. There are really 3 distinct types of drugs. They're administered by different routes of administration. Nusinersen is administered by intrathecal administration, omnisenvagine abaparavec by IV administration, and risdiplam by oral administration. They have distinct tissue and cellular distributions and really distinct molecular mechanisms of action. But all 3 of these work. And what's kind of striking from the clinical trials is that they're kind of more similar in their effects than they are different. So when given very early, they can be very efficacious, and when given later, much less efficacious. So just to remind you molecularly how these different drugs work, here is sort of the genetics of SMA again, usually large recessive deletion mutations of SMN1, but all patients retain at least 1 copy of SMN2, with the copy number correlating inversely with disease severity. So there are 2 basic strategies here. 1, replace what's missing. This is using a viral vector, adeno-associated virus 9, that contains a cDNA of the SMN gene. That's injected, gets into the spinal cord, into motor neurons, and makes full-length SMN RNA and full-length SMN protein. The other strategy is to ignore SMN1 and rather target the pre-mRNAs arising from SMN2. Now, SMN2 is very similar to SMN1, but it contains a nucleotide change, a cytosine to thymine change, that means that when these pre-mRNAs are made, most of the time, in the absence of treatment, they're spliced to remove an exon. The way these 2 drugs work, both nusinersen, an antisense oligonucleotide, and risdiplam, a small molecule, is that they encourage exon 7 retention, making full-length RNA and full-length protein. So all 3 drugs work to increase SMN protein expression. They just do so by different mechanisms. Okay, so I want to spend a minute just on each of these kinds of drugs because they're really fascinating, very novel, and rapidly being developed for multiple neurological diseases. So antisense oligonucleotides, these are chemically modified single-stranded DNA molecules. And they're composed of the antisense sequence complementary to a specific RNA. So really very highly specific, usually about 20 to 25 base pairs, and recognize a specific RNA with high specificity. They are taken up into cells via endocytic pathways and enter the nucleus. And some of these pathways, in fact, this biology is an area of active research because we don't understand these pathways all that well. But eventually the drug gets into the nucleus, and then it can bind RNAs and have several types of effects. You can engineer these drugs to either bind the pre-mRNA here, as is the case with SMN2, and by binding the pre-mRNA, you can alter its metabolism, you can skip an exon, or in the case of SMA, we're retaining an exon. Or you can actually engineer the ASO to bind an RNA and recruit an enzyme called RNase H, which then works to degrade that RNA. So in diseases where you have a gain of function, like Huntington's disease or SOD1-mediated ALS, you're using this strategy to actually degrade an RNA. But in the case of SMA, you're actually binding to an RNA and changing its splicing. So these drugs are, potentially have enormous potential ways that they can work. They do have some limitations. A big one is they don't cross the blood-brain barrier. They're big charged molecules. And so the way we currently deliver them is by a lumbar intrathecal injection. Okay, so nusinersen in particular, the ASO that's been approved for SMA, also the brand name Spinraza, it's an ASO that's chemically modified in a particular way. But importantly, what it does is it binds a specific sequence within the SMN2 pre-mRNA called ISSN1. The details of that don't matter. But the important point is that motif recruits a splicing factor that works to remove exon 7. So by giving this drug and binding this site, you sterically inhibit the binding of this splicing factor, and you allow retention of exon 7 and full-length SMN expression. Now, an important thing to consider in SMA is that we actually use a pretty low dose of this drug. We're only using 12 milligrams in 5 cc's, and we do that in a newborn infant all the way to a fully grown adult. And it's sort of an artifact of the original clinical trials where we were really targeting infants and wanting to make sure we had a safe dose and volume for infants. But importantly, we can probably go up on this dose, and it may be that a higher dose is more effective. And so there's currently a clinical trial going on called DEVOTE, which is testing a higher dose of nusinersen. This is just a slide that Wilden Farrell, who's no longer at Biogen, but lent me some years ago, that kind of makes the point about the potential efficacy of this drug, nusinersen. Some of this data was published in the New England Journal, but what you can see is that symptomatic SMA type 1 infants, these are infants who have 2 copies of the SMN2 gene. When you look at them on a motor milestone scale here on the y-axis, unfortunately, these infants never really achieve any meaningful motor milestones. They're profoundly weak. If you treat infants symptomatically after 6 months of age, when you start drug, they do clearly improve. But what's striking is if you treat infants less than 6 weeks of age, at a time when by pediatric neurologic exam we don't see any obvious deficits, you can see this much more impressive improvement in motor milestones. And this kind of pattern where we see efficacy in post-symptomatic treatment, but marked efficacy with pre-symptomatic treatment has been really replicated in all the clinical trials of risdiplam and omnisendigene abeparvec, the gene therapy. And this is just a video, maybe we can play the video, that just kind of highlights this effect. This was a sibling pair that my colleague Tom Crawford took care of. He took care of this little girl who had SMA type 1 prior to any treatment being available. And she is pictured, you can see at 2 months, very, very weak, and died by 15 months. But this is her brother. He also has 2 copies of SMN2, and he was treated with nusinersen at 3 weeks. And you can just appreciate the profound difference here already in his outcome. This is him at 15 months. He's able to crawl, a milestone you would never achieve in a type 1 infant. So it's really a remarkable outcome. And then here at 16 months, he's actually climbing stairs. So this, this is just almost miraculous, right? Really, really significant impacts. So where are we with other ASOs and neurologic diseases? There's so much activity in this space. This is a somewhat limited list, but I think it's important to have some perspective where we are. So there are some other ASOs or RNA-targeted therapeutics that are already approved. The other one that's most efficacious, and thus for you all to be most aware of, is 2 drugs that target the RNA in familial amyloid neuropathy. Now, this is a rare genetic neuropathy, but one that's really important to be aware of, because if you treat patients early, they can do very well. Now, this is less of a challenge in a sense in terms of drug development, because amyloid is actually transthyretin is the protein that forms the amyloid deposits. But this protein is actually synthesized by the liver. So when we're treating this disease, we're treating systemically and we're targeting the liver. And it turns out these drugs are robustly taken up by the liver. And so the actual drug targeting here is easier. The key is to recognize this disease and treat it early. There are other RNA targeting therapeutics that are already in the clinic for Duchenne muscular dystrophy. These are exon skipping drugs. These have less profound effects, sort of modest effects. And this may be because muscle biodistribution is actually kind of challenging. And there's a big effort going on to develop better drugs that can target the muscle more effectively. And you may have heard about the recent news regarding 2 ASOs, tominersen for Huntington disease, and then tofersen for ALS. And I'm going to touch on those in a minute. So what about this small molecule idea? So, I think when you talk to pharmacologists about the possibility of targeting a single RNA in the genome with a small molecule, they would have said that's impossible. How would you achieve the specificity to target this single RNA? But in fact, this ended up working in the case of SMA, that this drug, here depicted in this slide, actually binds the SMN2 pre-mRNA at 2 sites. And by doing so, sort of achieves a tertiary conformation that allows exon 7 to be retained. And it does so with very little off-target effects. There are a few other RNAs that can be impacted by this drug, and that does limit probably how high we can go on this drug. But this drug has been safe and well tolerated in clinical trials. It's now approved. And its huge potential benefit is that it's orally bioavailable. So patients take it as a liquid once a day, and it goes everywhere, not just the nervous system. There are 3 dose levels depending on the patient age, and there are some side effects in about 10% of patients. But overall, my own experience with this drug thus far is that it's been very well tolerated. Okay, what about onasemnogene abeparavec, or the trade name Zolgensma? So this is only the 2nd gene replacement therapy that's on the market now, that's actively being prescribed now. 3 total have been approved by the FDA, but 1 was dropped in commercial use because it just wasn't used much. 100 of these viral gene therapies are in clinical trials, just to give you some perspective of what's happening in drug development. So I thought it would be useful to just talk a minute. The adeno-associated viruses have really become the virus of choice for delivering genes. And in the case of SMA, as I've mentioned, it's driving a cDNA, but interestingly, it's using a constitutively active promoter and enhancer. So this is just kicking out SMN without any of the endogenous regulatory sequences. What's important about the adeno-associated viruses is that they do not integrate into the host genome. At least if they do so very inefficiently. So they remain as an episome, and they can therefore continue to make their product indefinitely in a non-replicating cell like a neuron. In replicating cells, this virus will get diluted out. But in a non-replicating cell like a motor neuron, these viruses can stay and express perhaps indefinitely. And this has led to the idea that this treatment could be one and done. Give it once IV to a small infant, and then you're finished. At this point, we can't redose these drugs because you develop AAV9 antibodies once you've been dosed with this that then preclude the ability to get it again. And if you have existing antibodies, in an infant, they often arise from mom and they will wash out over time. That may make you eligible to receive the drug, but if you have persistent antibodies, you're excluded from receiving the drug. Now, importantly, there is well-recognized short-term inflammatory responses. The liver sees a lot of this virus early, and so you have to dose with parallel steroids in order to keep that inflammatory response down. So this is data that from Jerry Mendel that show that at low dose of virus you don't have much effect on a motor functional outcome scale, but high doses of virus given once you see this marked improvement in SMA infants. So this drug, as we've mentioned, is already FDA approved. It was approved in 2019. It's approved currently in the US for infants 2 and less only, and one-time IV. There is an effort to develop intrathecal dosing for older age patients. That's in trial currently. So I'll stop there on that point. Okay. So I mentioned these short-term toxicities. One of the big ones is liver inflammation. So I am not aware of a child with SMA who's actually died of this liver inflammation. I'm aware of cases where very severe liver toxicity occurred, but all recovered. Most of the time, though, this can be managed readily with short-term parallel steroids. There's also a rare new side effect that's been recognized in the post-marketing era, which is called thrombotic microangiopathy. I think what the FDA is currently struggling with is how do we think about these toxicities that may arise from gene therapy, not just in the short term, but in the long term. How do we monitor patients? Because once this is given, you cannot withdraw it. So unlike risdiplam and nusinersen, where if you get into trouble, you can take it away, like we do with most of the drugs we prescribe in medicine, this one you cannot. Okay, there are, as I kind of mentioned, across medicine, 100 gene therapy products in clinical trial. These are some of the ones that are particularly ongoing in neurological diseases. Okay, so I wanted to touch on specifically the ASOs for ALS and Huntington's disease. So again, these are autosomal dominant disorders. Huntington caused by mutation, expansion mutation in the Huntington gene, and SOD1, one of the forms of familial ALS, is caused by dominant mutations of SOD1. So there have been recent trials with Huntington's disease trying to use an ASO to knock down Huntington, this delivered by intrathecal injection, and then an ongoing trial with tofersen, which is an effort to knock down SOD1 and ALS. Now, what about the trial for familial ALS? So this is tofersen. This is an ASO targeting SOD1. And patients were randomly assigned to receive tofersen by lumbar intrathecal injection or placebo. And then both groups, after 28 weeks, were rolled over into an open label study. And unfortunately, this was presented by my colleague Tim Miller at the ANA meeting. The announcement was made that at 28 weeks, they failed to meet their primary outcome measure. But when looking at the open label extension part of the study, you start to see some interesting patterns. So tofersen is reducing CSF SOD1 by about 30+%. Tofersen is reducing plasma neurofilament levels in the blood of these patients. And as you follow patients over time, there do appear to be some impacts on clinical outcomes. And some of those are just illustrated here. So you can see in the blue lines, those that were first enrolled in the placebo, and then here in the shaded area, both groups switch over to drug. And you can appreciate that in terms of neurofilament, respiratory function, clinical function, and time to event, that you start to see that there are some trends where these groups are separating. So this is somewhat promising that there is some effect happening with this drug over the longer term, but certainly this is not a resting disease. There continues to be progression in these patients. So what are the ongoing challenges? And I want to make sure I'm paying attention to time too. If you can interrupt me, Eddie, as soon as you want. But what are the ongoing challenges in developing these platforms for neurological diseases? Well, there are many things that come to mind for me include, molecular target engagement. How much up or down are we trying to manipulate this protein? And how the heck do we measure it? The nervous system is really tough. We really can't sample it readily in living patients. The best we have is CSF, and that might be quite distinct from the parenchyma of the nervous system. There are issues of drug biodistribution and potential toxicities. And then there's this really complex issues of the evolution of disease pathology. And what part of the cellular aspect of the disease is actually reversible or not? So coming back to SMA is due to reduced expression of this protein called SMN. And SMN is actually expressed in every cell in our body, so it follows this rule, seemingly, of neurodegenerative diseases, where it's a ubiquitous protein, but selectively affecting a particular cell type. This SMN complex, SMN interacts with a number of other proteins, and it's critical for forming what are called SNRPs. And SNRPs are critical components of the spliceosome. So SNRPs are critical for how genes are spliced. There's kind of an irony. The reason we have SMA is because alternative splicing of SMN2, but then the SMN protein product is critical for splicing. So it's thought that it's maybe splicing abnormalities that actually cause SMA, but we really don't know which RNAs and exactly how that works. We've always thought that too much SMN was probably fine, right? So some of these strategies, when we're targeting SMN2, we can't go above physiologic, right? Because we're limited by the template, the SMN2 RNA template that exists. But obviously, gene therapy, we can actually go above an endogenous level of SMN. And there's a recent study from colleagues at Columbia that suggests that at least in mice, when overexpressing SMN a lot, you can get a short-term benefit, but then long-term toxicity. So what's happened here is that mice now living, over a year are developing aggregation of the SMN protein in neurons, and this is making mice sick. So, this raises a concern. While we might have short-term benefits, what is going to be the long-term safety of some of these treatments? And Tom Crawford and I recently wrote an editorial sort of considering this. How do we do long-term safety studies in patients getting these drugs that may be in them for a lifetime in the case of SMA? So I made this point about how do we measure PK and PD in living patients. SMN can't be measured in the CSF, so we're even more limited than ALS or Huntington's disease. The only place we can measure it is in the blood, and here you can just see how variable it is between different people in the blood. It's pretty stable in an individual person, and thus when you treat with risdiplam, the drug that goes everywhere, we can monitor SMN in the blood and see it go up. We cannot do that with nusinersen or the gene therapy because these don't distribute into the blood. So this doesn't answer, though, even though we can see this level in the blood, what is happening with SMN in the CNS itself. And so what we did is to collect autopsies from patients. This took a long time. Each of these dots represent an individual patient between gestational ages into neonatal ages and afterwards. And this is looking at non-SMA patients at SMN levels in the spinal cord. And you can see how variable, but also how relatively high they are in utero relative to postnatally. And then if you look at SMA subjects, you can see that out here past 3 months of life, there actually isn't a statistically significant difference between SMN levels. But earlier, neonatally, and perhaps also in utero, these protein levels are different. So SMN levels are not static, seemingly, during development, and may be highest in utero. And it implies that maybe when SMN is really needed is when motor neurons are developing in utero. Okay, what about drug distribution in the CNS? So this is also data from autopsies that we collected, and we're measuring the level of nusinersen in different parts of the nervous system in patients that died of their disease. So of course, this is biased to those who did less well, but you can see, remember, it's delivered by a lumbar intrathecal injection, and not surprisingly, you have much more of the drug at lower lumbar levels, and then when you start to get up higher in the CNS, these levels drop down substantially. And this just emphasizes that's also the case when you do this by immunohistochemistry. This is looking at the drug itself in the spinal cord. Once you get up into the cortex, you really don't detect much drug except in sort of the meninges. This is looking what happens to SMN levels, and you can also see at the RNA level that SMN is going up in a caudal or rostral pattern. Interestingly, on a bulk level, bulk tissue level, we don't see an increase in SMN protein, indicating that overall the SMN protein induction is pretty modest in these patients. When we do immunohistochemistry for SMN, we do see a little bit of an increase specifically in motor neurons. So, this kind of work, working in autopsies, is so critical for understanding how these new drugs are actually working in people. And so I would implore you all, if you're taking care of these kinds of patients, please think about autopsies. What we're learning is that ASOs may be taken up with variable efficiencies by different cell types, and there's definitely a gradient, a drug concentration. And so the trial that I mentioned for SMA is a higher dose trial where 28 milligrams or 50 milligrams are being used. And importantly, in the ALS and Huntington's disease trial, higher doses of drug were used in order to try and get up into these deep, both superficial and deep cortical regions. You can see here when we measured in SMA-treated patients in these deep nuclei, we could see almost no drug, as shown by the yellow dots. We have much less data even with the other two drugs. So this is all the data we have for the gene therapy. This was an infant who died after receiving Zolgensma. They received drug at 2 months. What was done by Novartis and Arthur Burgess is to laser capture motor neurons from the autopsy, and maybe reassuringly, there was not an overexpression of SMN in the motor neurons in this case. If you do SMN staining, you can see an increase in SMN protein expression in motor neurons. But essentially, we have 1 or maybe 2 cases. And with risdiplam, we don't know anything. With risdiplam, we have no data about what drug levels or SMN induction is in the CNS. Okay, so I mentioned this issue that all 3 of the drugs are more effective when given very early. And this has led to the institution of population-wide newborn screening for SMA in the US. So now every infant, almost, just a couple of states lagging, but every newborn in the country is being screened for this disease at birth. And the reason for that is to identify infants very early and treat very early. It's challenging, though. What we're learning is even with newborn screening, we may not be getting in there soon enough in a lot of cases. So the emerging data is that about half of type 1 infants are symptomatic by the time treatment is started. And because of the challenges of these drugs, the cost of these drugs, amongst other things, most of the time we're not able to treat to about 3 weeks post-birth. Now that actually seems pretty fast. But what we're learning in SMA is probably every day matters. So why is this? What we think is happening, and again, this is work that we've done looking at autopsies, is that rather than just purely a neurodegenerative disorder, we think that this disease is really a developmental disorder in part. So here we're looking at a spinal cord isolated from an SMA patient, and the ventral root here. And you can see in this image how thin it is. It's like a thread. It's tiny. And if you look at it in cross-section compared to a control ventral root compared to SMA ventral root, you can see how small these ventral roots are in the controls, and that's just quantified here. And what we've learned when we look at the motor axons in the ventral root is that they're not developing normally, at least a subset of them. So if you think about normal axon development, they start off very small initially, they kind of directly abut each other, and so surrounded by the cytoplasm of a single Schwann cell, they grow in diameter, segregate one-to-one with Schwann cells, and eventually are myelinated. But what we see in human autopsies is that many of these axons are sort of stuck in an immature state. And that's also the case in SMA mice. This is the only SMA mouse picture I'm going to show you, but you can see here over developmental time that normal axons in mice are initially very small, grow, and eventually become myelinated. But in SMA mice, they're stuck in this very immature state and then die very rapidly postnatally. And we can monitor this degeneration using a serum biomarker. I'm sure all of you have heard about the emergence of following serum neurofilament in the blood. We hope that someday this is going to become the SED rate of the neurologist. When axons die, they release neurofilament proteins into the blood, and we can measure these in the blood. And when we look in SMA mice, we can see that their same level in utero, but at P1 and P2, they go sky high. So very early, there's this big release of neurofilament, indicating very precipitous degeneration. So what about patients? Do they show that same pattern? Well, the suggestion is that they do. This is what controls over life look like. There's a little bit of increase very early in life, probably normal developmental pruning kinds of events are driving that. When you look at pre-symptomatic infants with 2 copies of SMN2, you can see they're very high relative to controls. Those infants that are symptomatic with 2 copies of SMN2 are also very high, maybe a little bit lower. And then those with 3 copies are better off. And then as the infant ages, they actually fall rather than going up. So the highest levels of neurofilament levels in patients are right at the start, pre-symptomatic, before overt symptoms are seen. If you look with the drug at neurofilament levels, you do get a decrease in neurofilament levels compared to sham. So the drug is hitting target and reducing ongoing degeneration. How does this pattern of neurofilament compare to an adult onset neurodegenerative disease? So again, this is just a schematic, a cartoon showing you the infantile onset SMA. They're sort of sky high very early and then falling. Later onset SMA, they aren't as high, and that peak may happen a little bit later. ALS patients are sort of normal, and then at phenoconversion, they go up and they stay up. So a totally different pattern in SMA versus ALS. And then if you consider Huntington's disease, it's even harder, because what the Huntington's disease community is understanding is that this disease may have a very long prodromal phase. And if you look at neurofilament levels, you can see that they're starting to go up quite early. And this represents a huge challenge for Huntington's disease in terms of the timing of treatment. Okay, so I'm just going to conclude. We now have 3 gene-targeting therapeutics for SMA patients. Each of them increase SMN and ameliorate disease, sometimes markedly. I don't want to give you the impression, though, that this is a panacea. We see incredible variability in response. And when we give it later, there's much more modest improvement. And so we have much, much more work to do to optimize these treatments. Applying these strategies to adult onset neurodegenerative diseases, I think that what we're seeing in SOD1-mediated ALS is promising. And of course, now there's a pre-symptomatic SOD1 trial ongoing, and we might see much higher efficacy in that case. But clearly, it's going to be a little bit more challenging in these adult diseases. And, we really need to be very thoughtful about how we address these questions of, is the drug engaging target? Is the drug eliciting the expected biological response? And whether there's a clinically meaningful outcome. And then, because Eddie Patton is following me on this issue of cost, the elephant, of course, in the room about these drugs is their cost. So the New York Times wrote back in 2016, and actually the pop press mainly talks about the cost of these drugs, not the incredible breakthrough that they represent. But it's a really important issue to talk about, so I understand why that's made such news. But the drug called Spinraza will not come cheap, and by some estimates will be among the most expensive drugs in the world. So that was back in 2016. In 2019, when omnisemnogene obiparvec was approved, there was this quote from the Washington Post: "The costliest drug on the planet will treat infants with rare disease. The market fight focused on cost and safety is just getting started." So it costs $2.1 million for the single dose. It's one dose, but that's a price tag, obviously. And then risdiplam is the latest to be approved, and it's possibly a little cheaper than the other two, but still very expensive. So this creates huge issues, both in the US and around the world. Only about 40 to 50 countries of the 195 countries in the world have access to these drugs. There can be delays because of insurance approval, which obviously, as you can see, have huge impact potentially on long-term outcomes. So I'm looking forward to hearing Eddie talk about this issue a little bit further. And my last slide is just to thank people from my lab who contributed to some of the work that I showed today. Patients, this is a patient from when I was a fellow at NIH back in 2004, and happily gave their blood for an SMN and blood study. And my very long and close colleague, Tom Crawford, who he and I work very closely together at Hopkins. So happy to take any questions. Thank you.
Is a lab test for neurofilament currently available commercially?
It's not, but it's coming. So I think, we'll see. Probably the company that will get there fastest is Siemens. So they have developed a neurofilament light chain assay that can be run on their platforms, which of course are in many of the blood labs around the country. And at least last I heard, they're going to try and obtain approval for use in multiple sclerosis first. So that's probably where you see it first. The last I heard, it may be about 2-3 years away, but it has to go through a process of FDA approval as well. So we'll see how that goes. The challenge there is it's not very specific, right? Lots and lots of things can change your neurofilament level. And the other challenge, particularly for the SMA population, is though there's been thousands and thousands and thousands of normal adults profile for neurofilament level, because you have to establish a normal range of any blood test before it can be approved for use. But what's harder is the little kids. We don't know very much. That's the sum of the data I showed you today so we need much, much more data in small children for normal neurofilament level.
Another question, sorry my ignorance, but
No, not at all.
Is there a validity or use to screen moms during pregnancy so that you can start treatment as early as possible?
Yeah, thank you for that question. Actually, this funny. I took this slide out because it's a mouse slide, and I thought this was more useful to talk about human disease today. But yeah. Are we headed to in utero treatment for the most severe SMA infants? And there's increasing interest in that, particularly now that we have neonatal screening in place, and we're still finding that many of the kids are symptomatic by the time that we treat. So could you start in utero? And you can think about the 3 drugs and think about which one might be best in utero. Risdiplam has the advantage that it crosses the blood placental barrier. You could give it to mom. It could pass the placental barrier into fetus. And we showed in mice, at least, that this can work. The trouble is that issue of the small molecule potentially having off-target effects on other RNAs. So is it going to be safe in utero? That's the big issue. Viral gene therapy, I think, is the most scary for an in utero type of treatment paradigm. Antisense oligonucleotides, ironically, might be a good choice. They're pretty specific for a single RNA. The main issue there is how do you deliver it, because it wouldn't cross the placental barrier. But this is definitely an area of work, and SMA is such a great test case, because if it could work in SMA, it will really explode the field of in utero therapeutics, I think. So it's it's kind of an emerging field, which is very exciting.
Thank you.
Mm-hmm.
That sort of answer, or the question I was going to ask about this as a SMN being a developmental protein, because we really do see these therapies as remarkably effective, and although we don't see normal kids for the most part, we see pretty normal motor development. So is it a developmental protein? I think your data really does support that, but we still see efficacy with delivery after birth. So how do you resolve that, I guess?
So, I think you're getting at the basic biology. Do you need SMN only during development, or do you also need it for maintenance of the motor neuron? I don't know that question has been fully answered. The data that we have thus far suggests that you do need it both for maintenance of the motor neuron as well as the initial development of the motor neuron. But we've never probably fully experimentally restored normal development and then been able to withdraw and see. But it's a big question, and I think this will be a natural experiment that occurs. I think that, patients aren't always adherent to their medications, and I think we will see as time goes on, once the kind of child you're taking care of, Gary, who gets it really early, is doing pretty well, they're probably at some point going to stop on their own, and it'll be very important to see what happens. The other issue that we've struggled with as a field is do you need SMN replacement only in motor neurons? Or does the SMN deficiency that's actually present ubiquitously in the body have relevance at all? And some of the drugs obviously specifically target the CNS, whereas others are more systemic. To date, we haven't, in my opinion, seen really good evidence that SMN is critical in other tissues. But as these children live a long time, children with SMA1 who have relatively less SMN protein, as they live a long time, will we start to see phenotypes? And this is something really to be aware of as clinicians. Will we start to see phenotypes in other tissue types? And we just don't know. There's a lot we don't know. And this is why long-term follow-up studies are so key.
Agreed. We're certainly seeing that with Batten disease, with CLN2. So we've changed that to a chronic condition, but we're starting to see vision loss that we never really saw as initial presentations for
Right.
CLN2 disease. The other question I had was, I think you allude quite a bit to targeting therapies to, I guess, parts of the nervous system, with the suggestion maybe that it had something to do with the Huntington's disease trial, and we always wonder about that. And we know that there's flaws to intrathecal delivery for SMN, for instance, because some of those kids develop arachnoiditis, and that's been a problem in the field. But it makes sense that
pretty low frequency of that.
it is. But it happens. So there's probably no perfect delivery system, but would there have been a different outcome, you think, in Huntington's and other clinical trials if we delivered directly into the brain or to the target tissues?
Yeah, so, we just don't know the answer to that. And although apparently there was one autopsy that came out of the Huntington trial, I actually don't know the results of that to know, and I don't know that they've been publicly released, to know how much drug got into deep striatal regions and how much knockdown of Huntington was present in those regions. Yes, I'm throwing it out there, but you're right. We don't really know, certainly when there's a failure, how much of that is drug targeting, how much of that is target engagement, how much of that is toxicity. I mean, it's very hard to sort out when you can't sample the tissue in living patients. It's tough.
Or clinical design. I think that's something in neurology that we really haven't thought about, is how do you measure an outcome in these diseases? I like the neurofilament idea, but that's indirectly
it is indirect
to the clinical efficacy, the meaningfulness for that particular patient. And that population.
I mean, when talking to Tim Miller about the tofersen trial, he now, though there have been so many clinical trials done in ALS, lots of practice there now to do a clinical trial, in retrospect, really wish that it hadn't been a 28-week primary outcome measure. So, we learn every time we do one of these studies with every new kind of therapeutic platform.
Fantastic talk. Thank you.
Thank you, Gary.
Hi, Dr. Sumner, a wonderful lecture.
Please call me Charlotte.
I did 2 years of neuroscience research. So we were using small hairpin RNA and using plasmid as a vector. And we're using lipofectamine, trying to infect the ex vivo rat hippocampal cells. So one difficulty was that it's so hard to transfect hippocampal cells or any neuron cells compared to other cell lines. And our lab spent a lot of time to find the optimal technique to transfect the cells. And then we developed the protocol, and the transfect rate was around 2% or 5%. And then we're measuring the expressed protein. So one thing is that we're considering the transfected cells a little bit less healthier than the non-transfected cells because their cell membrane was damaged a little bit to allow the DNA in. The second thing is that even it's transfected, the decrease of that protein expression is not very remarkable. I wonder if it's the same case in vivo cells for neuron cells.
So you're right. I mean, primary neurons are notoriously difficult to transfect with lipid-based techniques. So the remarkable thing about ASOs and the gene therapy is that they get taken up by neurons really readily. Exactly how that occurs is quite variable, probably cell type to cell type. But when you look in the humans, the ASOs are best taken up by neurons. So neurons really like them. Liver cells also really like them. Glial cells maybe a little less. So understanding those endocytic pathways better about how they're taken up and in different cell types how they're taken up is really important. Similarly with the viral vector, exactly how it passes the blood-brain barrier, how it's taken up into neurons. These are really interesting questions that are going to allow us to make these drugs better. In terms of the question is there data to suggest that the uptake of the drugs would vary depending on the health of the cell? It's definitely an idea that I've had. We have not in human tissue or even in mouse tissue really addressed that. There isn't obvious data from what we have so far that it's markedly variable depending on, in a given motor neuron population. But these are things we have to understand, absolutely.
Thank you.
All right. Thank you, everybody.